we are going to see where does photosynthesis take place in plants. Okay. So here photosynthesis generally takes place in green leaves. Okay, green leaves of plants. Okay. It also takes place in the other green parts of the plant, like green stems and everything. Okay. So here it generally the photosynthesis occurs in the mesophyll cells in leaves. Okay. So these mesophyll cells we have already seen in the anatomy of flowering plants. There will be in mesophyll there are spongy parenchyma, parasite parenchyma, it will be arranged. So mesophyll cells have a large number of chloroplasts. They have many number of chloroplasts. These chloroplasts are arranged. They are attached to the surface, inner surface of the mesophyll cells. Okay. They are attached to the surface. Okay. So when they attach nearby to the surface, when the light intensity or the light falls on the leaves, it can absorb the light energy. These chloroplasts. Okay. So these chloroplasts contain grana, stroma lamellae, and fluid stroma. So grana is nothing but the thylakoids. Each thylakoid is known as grana, and the stroma lamellae is nothing but the connecting between one grana and another grana. This one stroma lamellae. And the stroma fluid is nothing but the fluid in this. This fluid is known as stroma fluid. Okay. So here this system is responsible for trapping light energy and also for the synthesis of ATP and NADPH. ATP is nothing but adenosine triphosphate. Okay. So by trapping light energy, NADPH it is also a chemical compound like ATP. Okay. So here, so when the light falls on these mesophyll cells, so it absorbs the light energy and produces these chemical bonds, ATP and NADPH for energy. These are known as energy bonds. Okay, they prepare these energy bonds. So this process is known as light reaction because it is dependent on light. Okay, light reaction because it traps light energy and it prepares energy bonds like ATP and NADPH. This is known as light reaction. And after some time in the stroma, okay, stroma is the fluid here. In the stroma, what does this stroma do? It incorporates CO2. It absorbs CO2 into the plant, and with the help of CO2, it synthesizes sugar. Yes. Okay. Sugar is carbohydrate C6H12O6. To form starch. Okay, it synthesizes sugar and forms starch. So this reaction, which is incorporating CO2 and synthesis of sugar starch, this reaction is dependent on the chemical bond, energy bond. With that energy only it traps CO2 and synthesizes sugar and starch. Okay. So since this is not directly dependent on light, so it's called a dark reaction. Yeah. See, you clearly understand this. This process is not directly dependent on light, but it depends on light products like ATP and NADPH. Okay, it is dependent on the products of light reaction. Okay, so therefore it is known as dark reaction. So this dark reaction does not mean that this process takes place only in the dark. Okay, it takes place on the light also, but it does not depend on the light intensities. Okay, so therefore it is known as dark reaction. Okay, can you understand what is light reaction and dark reaction? So light reaction is absorbing light energy and transforming into chemical energy bonds ATP and NADPH. This is dependent on ATP and NADPH, but it is not directly exposed to light. Yes, you know it is exposed. It is not exposed, but it does not need light. Yeah, does not require. This is where only depend on light products. Yes. Okay. So next we will see about how many pigments are involved in photosynthesis. How many pigments are involved? That is our next topic. Now we are going to see what are the pigments involved in photosynthesis. Okay. So leaf pigments are separated. There are many pigments in leaf. So these pigments are separated through paper chromatography technique. Okay. So various pigments are arranged in paper chromatography techniques. If you take chlorophyll A, it is observed in the bright or blue-green regions in chromatography. Okay. So it is observed in blue-green regions of chromatography. Okay. So if you take chlorophyll B, so it is observed in yellow-green regions of chromatography. Okay. And xanthophylls are observed in yellow region. Carotenoids are yellow to yellow-orange region. So these colors are very important, which are observed in the chromatography. Okay. So here chlorophyll A is bright or blue-green in chromatography. Chlorophyll B is yellow-green. Xanthophylls yellow, carotenoids yellow to yellow-orange. Okay. So here these pigments are they differently absorb the wavelength. Okay. Wavelength of absorption of light. So here in that case chlorophyll A, it absorbs the wavelength of blue and red regions of light in the prism. Okay. We have already seen this one in classes. It absorbs blue and red regions of the wavelength. Okay. So we know that red color have long wavelength. Okay. So here, since chlorophyll A is associated, it is observed in the blue and red regions. It is known as chief pigment. So it has a very high wavelength and it is known as chief pigment in photosynthesis. Okay. So the other pigments like chlorophyll B, xanthophylls and carotenoids. So these other pigments are known as accessory pigments. So chlorophyll A is a chief pigment and the other pigments are known as accessory pigments. Okay. So what these accessory pigments do? So chlorophyll A pigment is the center. It is like a head of these pigments. So the other pigments are known as accessory pigments and these accessory pigments absorb light and transfer it to chlorophyll A. Okay. So these transfer the energy to chlorophyll A. Okay. So and these accessory pigments, they also protect chlorophyll from photooxidation. Okay. So what is
photooxidation?
Yeah, photooxidation is nothing but there occurs some oxidation during photosynthesis.
Exchange of gases.
During photosynthesis by the electron transport and everything. So it has a cause of damaging the pigment. Okay. So these accessory pigments protect those pigments from damaging.
Okay.
Okay. So it also transfers whatever the light energy it has absorbed to chlorophyll A. Okay. So these accessory pigments are surrounded to chlorophyll A, which is chief pigment. Okay. So these are the pigments involved, and the absorption spectrum we have studied here. So next we will see what is light reaction. Okay, light reaction we have already seen in the last class that which traps the light energy and produce it to ATP and NADPH bonds. Okay, so we'll deeply see what is light reaction. Okay. Now we are going to see generally about what is light reaction. Okay, we are not going in detail in this topic. We are going to see generally what is going in light reaction. Okay. So here this light reaction, what all happens in light reaction? First we have to understand what all the process happens in light reaction. So here it absorbs light. Light absorption takes place. Water splits into hydrogen and oxygen. So splitting of water takes place here. Oxygen released. Formation of high energy chemical bonds like ATP and NADPH. So this we have already seen, but we are seeing the functions of what are going on in light reaction. Okay, light absorption, water splitting, release of oxygen, and formation of chemical bonds like ATP and NADPH. Okay. So here generally pigments. Okay, what are the pigments? The pigments are grouped into two complexes. Okay, photosystem 1 and photosystem 2. So these are two complexes where
Photosystem or photosynthesis?
Photosystem. Okay, photosystem 1 and photosystem 2 because it is a complex. Okay. So in this light reaction, photosystem 1 and photosystem 2, right? So here these two are named according to the sequence of discovery. Photosystem 1 was discovered
first and
photosystem 2 was discovered. So there are in each system there are hundreds of pigment molecules. Okay, pigment molecules like chlorophyll A, chlorophyll B, xanthophylls and everything. They are bound to proteins. These all pigments are bounded to proteins. Okay, both the photosystems 1 and 2. So here a single chlorophyll, okay, it acts as a reaction center. We have already seen chlorophyll A is a chief pigment. So it acts as reaction center. Okay. So this photosystem, why it is differentiated into photosystem 1 and photosystem 2? It is because in photosystem 1, the reaction center absorbs the wavelength of 700 nanometers. Okay, it absorbs 700 nanometers.
In photosystem 2, it's 680.
80 nanometer. Okay, it absorbs the light at 680
20 nanometers difference.
Yes. So photosystem 1, you have to remember very well. We have to remember this. Photosystem 1 is for 700 nanometer. And photosystem 2 is for 680 nanometer. Okay. So maximum absorption takes place at 680 nanometer. Maximum absorption takes place at 700 nanometer. Okay. So it is a wavelength. Okay. So here since it is maximum absorption takes place in 700 nanometer, it is known as photo P700, and here it is known as P680. Okay. So these are general things about light reaction. What is happening in light reaction. Okay. So in the next topic we will be discussing about the electron transport. Okay, how the electron is transported and everything will be discussed. Why the electron should be transported. So everything will be discussed in the next topic. Now we are going to see about the sixth topic, which is electron transport. Okay. So here there are two light harvesting complexes. We have seen that already. Two photosystems. Photosystem 2, photosystem 1 and photosystem 2. Okay. So here what happens here is generally the electron transport, how it occurs means. So this is a light harvesting complex. This is another light harvesting complex. This is a photosystem 2, which absorbs light of wavelength 680 nanometers. Okay. And this is a photosystem 1, which absorbs light of about 700 nanometers.
Yeah, that we are already seeing.
So here what it happens, right? First when light falls on a leaf, so this photosystem 2, which absorbs light at 680 nanometer, it absorbs a light. Okay, it absorbs a light, and through that energy, electrons excite in the stage in this light harvesting complex. Electrons excite, and these electrons are moved to electron acceptor.
Yep.
Okay. So after moving into electron acceptor, this electron acceptor passes to an electron transport system. Okay, which is made up of cytochromes. Okay, electron transport system is made up of cytochromes, and it passes through photosystem 1, light harvesting complex of photosystem 1. Okay. So again these electrons are excited by light, and they reach another electron acceptor. Okay, and while reaching after reaching this another electron acceptor, it is joined with NADP plus to make it NADPH. Okay, so with NADP plus, if an electron is added, it becomes NADPH. Okay, and energy is produced here. So here this is known as Z scheme actually, because of the diagram actually. So because of the scheme of the electrons traveling, it is known as Z scheme. The Z
You can also call it as N scheme.
So they have kept it is also, this is also forming Z, right? So they have kept it Z scheme. Okay. So we cannot decide the scheme. They are only decided. So after that, so this is the electron transport system. How the electron is transported. Okay. So the water splitting complex is in photosystem 2. Here only water splits. That is water splits into 2 electrons plus 2 protons plus oxygen. So this oxygen is released. So this electron only is moved. Okay. After moving of electrons, there should be electrons to produce light. So it comes from water. Okay. So now we will see the splitting of water. The electrons replaced. The electrons which are replaced by these electrons replace the electrons which are already present in the light harvesting complex of photosystem 2, which are of wavelength 680 nanometer. So these electrons are replaced through splitting of water. Okay. So we have already seen this reaction, splitting of water. It produces H plus, a proton, oxygen, and electrons. Okay. So this is the reaction, and this mainly occurs only in photosystem 2 and not in photosystem 1. Okay. It only occurs in photosystem 2. Okay, these are water splitting complex. So here this water splitting complex, it is present in the inner side of the thylakoid membrane. Okay, it is present in the inner side of the thylakoid membrane. So this is about splitting of water. So next we will see cyclic and non-cyclic photophosphorylation. Okay. So now we are going to see about cyclic and non-cyclic photophosphorylation.
Okay.
So here, what is photophosphorylation? So photophosphorylation is nothing but the process of which ATP, adenosine triphosphate, is synthesized by cells like mitochondria and chloroplast in the presence of sunlight. Okay, it is known as phosphorylation. So in the presence of sunlight means it becomes photosynthesis. Okay. So generally preparing ATP and NADPH bonds is phosphorylation. Okay. In the presence of light energy, it is known as photosynthesis. So here the two systems, okay, the two photosystems PS1 and PS2, like we see in the last class, photosystem 1 and photosystem 2, by electron transport chain the electrons are passed away. So that when these two photosystems work in series, that is first photosystem 2 and then photosystem 1. So this kind of photophosphorylation is known as non-cyclic photophosphorylation because the electron is not cycled back. It is used up for to produce NADPH. Okay. So here ATP and NADPH both are synthesized by this non-cyclic photophosphorylation. Okay. So in cyclic photophosphorylation is when the electron from the chlorophyll is excited and again it is come to the same chlorophyll. It is known as cyclic photophosphorylation. So here when only one photosystem is functional, cyclic flow occurs. Okay. So that photosystem is PS1, which absorbs the light of wavelength 700 nanometers. Okay. So from here the electron excites and it enters to electron acceptor.
Okay.
And this electron acceptor releases electrons to electron transport system. So this electron transport system converts adenosine diphosphate plus inorganic, it adds inorganic phosphate to adenosine diphosphate and produces ATP, that is adenosine triphosphate. Okay. So here only ATP is synthesized, not NADPH. In cyclic photophosphorylation, only ATP is synthesized. Whereas in non-cyclic photophosphorylation, both ATP and NADPH plus H plus is synthesized. Okay. So where does cyclic photophosphorylation occurs? So cyclic photophosphorylation occurs in stroma lamellae.
Stroma lamellae.
Yes, because it does not contain photosystem 2. It only contains photosystem 1. Okay. So next we go to the next subtopic, which is chemiosmotic hypothesis. We are going to see about chemiosmotic hypothesis. Okay. So here this hypothesis explains that how ATP is synthesized. Okay. So generally plants synthesize ATP by electrons, we know that generally. So how this process is going on, we are going to discuss here. So here development of proton gradient occurs across the membrane. Okay. Membrane of thylakoid. So why it is proton is accumulating there in the membrane of thylakoid, inside the lumen of thylakoid. So we are going to study what are the reasons for proton accumulation inside that. Okay. So proton accumulation in the lumen of thylakoid. Okay. So here reasons for proton accumulation. Okay. So here first one, there are three reasons totally. The first one is splitting of water in the inner side of the membrane. So this is the thylakoid membrane. Okay, thylakoid membrane of grana. So this is the thylakoid membrane of grana. This is outer membrane and inner membrane. Okay. So here only photosystem 2 is present. Okay. So inside this photosystem 2 only water splits into oxygen, electron and proton. So electron is transported, it excites and it is transported to photosystem 1 and again it is and again it excites to produce NADPH. Okay. So electron does this function. And oxygen is released out. So there remains only proton inside this thylakoid membrane. So it is one cause of accumulation of protons inside the lumen, inside the thylakoid lumen. And the second one is the primary electron acceptor is a hydrogen carrier, is a proton carrier. Okay. So this from PS2 photosystem 2, when the sunlight fall on photosystem 2, here electron is excited from the splitted water. The electron is carried and is excited, and it reaches the primary electron acceptor. So when it reaches this primary electron acceptor, it carries a hydrogen from the stroma into the lumen. Stroma is the outside fluid present in the chloroplast. So it carries protons from there to inside the lumen. Okay. And then by carrying that one, it transfers the electron to photosystem 1.
Okay.
So therefore the protons accumulate here. Okay, when it carries the electron, proton is carried from the stroma into the thylakoid lumen. So it is the second cause of proton accumulation inside the lumen. Okay. So and the third cause is the Madp reductase enzyme. Okay, it removes protons from stroma. Actually, the electrons reach photosystem 1. By getting those electrons and protons from these stroma, NADP plus is changed to NADPH. Okay, so that chemical bond is created. So how is ATP synthesis? Therefore, the proton concentration in the stroma gets decreased. It is reduced. So therefore, proton concentration inside the lumen is increased. So this favors the proton gradient inside the lumen and outside the lumen. So with this proton gradient, which the more protons accumulate in the lumen, so this is accompanied by ATP synthesis. ATP synthesizing. Okay. So how ATP is synthesized? So there are some membrane proteins which are F0 and F1. So these help to synthesize ATP. Okay, we will be studying in the next slide.